Chapter 22, The Immune System, Part 1, Screencast 2. Now, the first type of innate immunity that we will be addressing are those that work by preventing entry into the body. Probably one of the best defense systems that we have is our skin. Very few microbes have the ability to penetrate skin while it is intact. It acts as a physical barrier. Remember, the skin is made up of the epidermis and dermis, but also it's just not that. The skin releases many antimicrobial substances. Examples would be dermacidin, lysozyme, sebum, and defensins. Skin also has a normal non-pathogenic flora, and so these are going to be microorganisms, and their role is to prevent the growth of pathogenic microorganisms. We also know that we have mucous membranes that line body openings. They produce mucus, and they also release antimicrobial substances such as defensins, lysozymes, and antibodies. They are also lined by harmless bacteria that will suppress the growth of more aggressive or pathogenic bacteria. Now here, we're focusing on different types of cells. And the first that we're going to look at, those cells that work through the process of phagocytosis. Cells that work in this particular way are neutrophils, macrophages, and dendritic cells. And once again, their mechanism is through the process of phagocytosis. And remember, that is a subcategory of endocytosis, and that is vesicular transport into the cell. So here we basically see a dendritic cell. We also see a macrophage. Remember, these are cells that work under this particular mechanism. And so notice we have an infectious agent and it's actually being engulfed. And notice what happens. It creates invagination within the cell membrane, and the membrane will close behind it, creating a vesicle known as a phagosome. That phagosome will be presented to a lysosome. Now we have a phagolysosome, and this is where the infectious agent will be destroyed, and any leftovers from the destructive process will be removed out of the cell via the process of exocytosis. There are also a number of pro-inflammatory chemical secreting cells. Examples would be basophils and mast cells. And these are cells that release substances such as histamine. This is a very potent vasodilator, and it also increases capillary permeability. There is heparin, which is an anticoagulant, and there are also eicosanoids, and these are going to be agents that actually increase or induce the process of inflammation. There are some cells, such as natural killer cells, that actually work through apoptosis. And apoptosis is actually cell death. So here we have a natural killer cell, and notice it is actually releasing two different substances. One is perforin, that is actually a substance that will create a transmembrane pore in the unhealthy or wanted cell. Then there's something known as granzyme. Granzyme will enter the unhealthy cell through the perforin, and then granzymes will cause apoptosis of that cell. We have eosinophils, and eosinophils are specifically white blood cells that work or release cytotoxic chemicals that are going to destroy or kill parasitic worms. Now here we have interferon, and one of the things that I want to point out here, you are not responsible for the subtypes of interferon. That's going to be important. What we do want to know is that interferon is primarily involved in viral infections. So let's take a look at what's happening here. There's going to be two general mechanisms that we are going to see here in which interferon works. Now, we're starting off with an infected cell, and notice it's actually releasing a number of interferons. And it's going to have different jobs based on the type of cell. When we look at a normal cell, interferon will basically synthesize enzymes that will interfere with the replication of the virus. Remember, viruses require a host, and they cannot replicate themselves without a host. Now, the other thing that they can actually do is result in activating natural killer cells and this will result in apoptosis, or they can actually activate macrophages, which will phagocytize infected cells. We also have a group of proteins known as complement. This consists of over 30 plasma proteins, and keep in mind the word complement, to work with. So complement systems are these plasma proteins that actually will work in conjunction with antibodies. They are identified by the letter C and number. These are proteins that are continuously synthesized by the liver. They are released in a form that is not active, and activation will occur through an enzymatic cascade. One thing to remember about complement, they are especially potent against bacterial infections. So remember, interferon works primarily with viral infections, whereas complement will be working primarily against bacterial infections. Now, if we look at activated complement, there are three general processes that we're going to see, or four, excuse me. One is opsonization, and this is when the complement protein binds to a pathogen, and what it does, it's tagging it. It enhances the likelihood of phagocytosis of that cell that is pathogenic. Inflammation is also enhanced by complement, so this will activate mast cells and basophils, also attracting neutrophils and macrophages. Cytolysis, complement triggers the splitting of the target cell. Complement proteins form these membrane attack complexes, or MACs, that create channels in the target cell, and basically fluid enters that cell, causing the cell to lyse or die. There also is the elimination of immune complexes. Complement links antigen-antibody complexes to erythrocytes. Cells move to liver and spleen, where the complexes are stripped off and removed and destroyed. Here we can actually see this. So kind of think of opsonization and notice that the pathogen has been tagged. And that's a really good way to think about opsonization. So now you've tagged this particular pathogen and kind of indicated that this is a cell that needs to be removed or destroyed. The macrophage recognizes that and will take care of that process through phagocytosis, killing the particular pathogen. If we look at the other role of increasing the inflammatory response, so remember, complement will activate and attract various innate immune cells. Examples would be macrophages, neutrophils, basophils, and mast cells. If we look at cytolysis, here we can basically see the complement protein creates a substance that actually will cause the cell to lyse. And the last method, remember, was elimination of immune complexes. So here we actually see an antibody. The antigen is bound to the antibody, but we also have complement, which cross-links with a red blood cell. And now that will be transported to the liver and spleen, where it will be removed.